Hello everyone. This lecture will be on classification of amino acids. In the last video we discussed about its introduction, and in this lecture we will deal with its classification, chemical nature as well as its biological role. Let's get started. Classification on the basis of the composition of the side chain or the R group. So this R is a side chain. This can be anything. This is different to each amino acid. It can be simple hydrogen in case of glycine. It can be methyl in case of alanine. It can be this entire R in case of leucine, and it can be this in case of isoleucine, etc. So this R is not unique to each amino acid. So each amino acid has different side chain. That's it. And as you can see here, there is an alpha. What is this alpha? In organic chemistry, alpha carbon refers to anything that attaches to the main functional group. How to determine which one is main functional group? In the last video we discussed regarding this. Actually there is an IUPAC rule. In that order, the carboxylic acid comes first and the amine comes last. So carboxylic acid act as a main functional group and amine here act as a substituent. So this alpha carbon in amino acid attached to the main functional group, which is carboxylic acid. What is then beta and gamma carbon? The beta carbon, if at all present, will attach next to this alpha carbon, and gamma carbon will attach next to this beta carbon. That's it. So on the basis of the composition of the side chain, amino acids are classified into following categories. First, having aliphatic side chain. Aliphatic refers to anything that is non-ring. It can be straight chain or branch chain, but it should not possess any ring like structure. So non-ring hydrocarbon chain, we call it aliphatic chain. Non-ring hydrocarbon chain, aliphatic chain. So for example, glycine, alanine, these are non-ring side chain. There are no benzene like ring present. That's why these entire compounds come under aliphatic side chain amino acids. Next, amino acid with an aromatic side chain. Aromatic refers to possess a benzene like ring. Possess a benzene like ring. That's why we call it aromatic side chain. Next, amino acid containing a hydroxyl group. Now here it contain a hydroxyl group, here it also contain a hydroxyl group. And this hydroxyl group are a part of the side chain R. This hydroxyl group is a part of the side chain R. Got it? Clear? Next, sulfur containing amino acids contain a sulfur atom in its side chain. Contain a sulfur atom in its side chain. Next, acidic amino acids. Acidic amino acids refers to it contains a carboxylic acid group in its side chain. Contains a carboxylic acid group. We all know that amino acid contains a carboxylic acid, but acidic amino acid means additional a carboxylic acid group is present in the side chain. That's why we call it acidic amino acids. A additional carboxylic acid is present in side chain. Got it? C. There are many structures, but do not worry. These structures are just for your understanding. You do not need to write those structures in classification part. Those I have made for just your understanding only. Got it? Next, move to basic amino acids. Basic amino acids, it contain a amine group. Why amine is basic? Because nitrogen, there is a lone pair, and this lone pair will accept a hydrogen, a H plus, a proton it will accept. That's why, since a nitrogen having a lone pair will accept a proton, according to Bronsted-Lowry theory, it is a base. That's why it is called as basic amino acids. Basic amino acids contains a amine, and because of this nitrogen it will accept a proton, because nitrogen having a lone pair. That's why we call it basic amino acids. What are amino acid amides? Look, these are the compounds, they are derivatives of these acidic amino acids. What happens here is this carboxylic group converted to amide group, CONS2. Converted to amide group. That's why amino acid amides. Clear? Next, heterocyclic amino acids. Heterocyclic ring. Heterocyclic ring refers to any atom, refers to carbon is replaced by any atom. It can be oxygen, it can be nitrogen, it can be sulfur, it can be anything. So there is an atom which replaced the original carbon atom in the ring. That is heterocyclic ring. So here, as you see on your screen, nitrogen replaced the carbon. So these are the heterocyclic amino acids. Clear? On the basis of composition of the side chain, up to these are the subcategories. Next, on the basis of nutritional requirement. Two types, essential, non-essential. What are essential amino acids? Essential, non-essential. What are essential amino acids? Essential in the sense they are essential for these functions. They are essential for these functions. That's why they are called essential. And another thing is that these amino acids are not synthesized by human body. So you must get it from diet. That's why they are essential amino acids. They are not synthesized by human body. You must get it from diet. That's why they are essential in nature. Clear? So there are 10 essential amino acids and 10 non-essential amino acids. Non-essential are those that are synthesized by human body. So you would not need them from diet. Essential refers to they are not synthesizing by your human body. So you must get it from your diet. Clear? Next, on the basis of polarity of side chain. Now whether the side chain attached, the R group attached is polar or non-polar. What is polarity? Polarity depends on two things. One is the electronegativity. Listen carefully everyone and look at your screen. Polarity depends on two things. One is electronegativity. Another one is shape of the molecule. Shape of the molecule. What is electronegativity? Electronegativity depends on multiple factors. First, there should be small atomic size. Atomic size should be smaller. What will happen? These smaller atoms will help attract electrons because of their close vicinity to nucleus. So the smaller atomic size, greater electronegativity. Next, number of protons. Look at here. Helium is super small, but helium is not more electronegative. Why? Because all these factors must consider. A atom should possess all these factors to classify into electronegative. Next, number of protons. More number of protons, more electronegative. Organsen, atomic number 118, but it is not more electronegative. Why? Because its atomic size is bigger. It is a bigger atom. So all these factors should possess one particular molecule, particular atom to classify into electronegative. Next, shielding. What is shielding? Look at closely here. Let's say this is a nucleus. A nucleus contains protons. Protons are positively charged particles, isn't it? So around nucleus, the 1s cell is there. Then there is 2s and 2p cell are there. 2s 2p, isn't it? Let's say there are 2 electrons in the 1s cell. There are 2 electrons in the 2s cell and 4 electrons in the 2p cell. Why I am drawing 2s and 2p on the same? Because the quantum number is 2. Because the quantum number is 2 of the second cell, 2s 2p. Quantum number is 2, 1s quantum number is 1. What shielding is actually? This is a positively charged proton present here. Proton is present inside the nucleus. Proton is positively charged. This positively charged proton will attract electron. This will attract electron from its valence shell, the outermost shell. It will attract electrons. It will attract this electron. It will attract this electron. It will attract all those electrons present in the valence shell. But what happens is, in the valence shell there are 1, 2, 3, 4, 5, 6 electrons present in the valence shell, but in the inner shell there are 2 electrons present. What these 2 electrons will do? These 2 electrons will prevent any of these outermost shell electrons from reaching towards the nucleus. These 2 electrons will shield the nucleus, will prevent the nucleus to get these outermost electrons. Because this is negatively charged, this is negatively charged, this is negatively charged, this is negatively charged. These 2 electrons will repel these 2 electrons. So these 2 electrons will not get attached to the nucleus, to the proton. This proton will attract these 4 electrons only. These 4 electrons only. Here shielding is low. The less the shielding, the more electronegativity is. Let's me drawing first. There is a nucleus. Protons are present. There is a first shell and there is a second shell. This is 1s and this is 2s and 2p. Since there is a innermost electrons are there, these innermost electrons will prevent the outermost electrons from reaching towards the nucleus. The less the shielding is, the more electronegativity is. Why? Because these positive charges present inside the nucleus, these protons will attract electrons negative, protons positive. So the more number of electrons it will attract, the more electronegative that atom is. Let's say, for example, let me draw again. Now this is proton present inside nucleus. This is first cell and this is the second cell. So there are 2 present in the first cell. There are also only 2 present in the second cell. 1s2, 2s2 and 2p2. So these innermost electrons will prevent these 2 electrons from reaching towards the nucleus. Here electronegativity is low compared to the atom having outermost 6 electrons. Clear? The less the number, the less the shielding is, the more the electronegative the compound is. Here these 2, here suppose there is a presence of 4, here 2, 1, 2, 3, 4, 5, 6. It is more electronegative than this. Why? Because there are 6 electrons present in the outermost shell. Here 4 electrons present in the outermost shell. But out of 4, only 2 are reaching towards the nucleus. 2 are blocked by these innermost electrons. But here 4 are reaching towards the nucleus. 2 are blocked by this innermost electron. So this is more electronegative than this. Clear? This is known as shielding effect. Shielding effect. The innermost electrons prevent reaching valence electron towards nucleus. This is called as the shielding effect. The less the shielding, the more electronegativity. Another factor that is not very important but is significant is lone pair. More lone pair, more electronegative. Fluorine has 3 lone pairs. Oxygen has 2 lone pairs. Fluorine is more electronegative than oxygen. So looking at all those factors, fluorine is more electronegative than oxygen. Oxygen is more electronegative than chlorine. Like this. Clear everyone what is shielding? Shielding in simple language refers to how much number of electrons are attaching with nucleus. If there is a outermost shell 6 electrons present and in another atom the outermost shell is 4 electrons present, the 6 electrons present will is more electronegative because 2 electrons only blocked by those innermost electrons. That's it. Clear? Another, this is all about the electronegativity. Then there is a shape of a molecule. Shape of the molecule. What is that shape of the molecule? Whether the molecule is linear shape or bend shape, what will happen is, here look at the water. Water is more electronegative. It will attract this shared pair of electron towards itself. Since it will attract shared pair of electron towards itself, the oxygen becomes partial negative charge, the hydrogen becomes partial positive charge. So one end is partial negative, other one end is partial positive. That's why water is polar in nature. Because this negative part will attract a positive part and this positive part will attract a negative part. So these water molecules hold together. But in case of here carbon dioxide, oxygen, it will pull share electrons towards itself. The one end is negative charge, other end is negative charge. Both ends are negative charge, it cancel each other. Because in water, one end is negative, other end is positive charge. So one end is negative here, it will attach to the positive charge, but the other end is also negative here. And this water is other end is negative here. This will repel, this will not attach to each other. That's why carbon dioxide is not polar. Because of the shape, because this shape. Here water is in bend shape. That's why this is the one end, this is the other end, and one end is negative, other is positive, the opposite. But here both ends are same. Both ends are same. This water is bent because of electron repulsion. Had this carbon dioxide is bent, this also becomes polar in nature. Look at another example of ammonia. Ammonia, look at this, triangular in shape almost. Nitrogen lone pair. Here it will slightly negative, slightly positive. This slight negative part will attach to slight positive part of water, and this positive part will attach to slight negative part of water. So this is soluble in water, this is polar in nature. Ammonia is polar in nature. Slight negative, slight positive, polar in nature. But here both are negative, both ends are negative. That's why it is non-polar in nature. Clear? So polarity depends on two factors as we discussed now, electronegativity and shape of the molecule. So based on polarity, amino acids with non-polar side chain. Why non-polar? Because carbon, hydrogen are non-polar. Why? See, though carbon is more electronegative than hydrogen, their difference is too small. They almost pull equally. So since their difference is very minimum, so they pull equally, that's why they are considered as non-polar. The carbon-hydrogen bond considered as non-polar. So these are the compounds having non-polar side chain. Next, amino acids with polar but uncharged R groups. Look at this, polar because there is oxygen present. Oxygen is more electronegative, more polar. All are oxygen present, so there is a polar in nature. But uncharged, there are no charge here. There are no charge like positive or negative charge are present. That's why they are regarded as uncharged side chain. Clear? Next, amino acids with negatively charged. Negatively charged means here COO minus is there. Here also COO minus is there. That's why negatively charged R groups. This is the R group, this is negatively charged. This is the R group, this is negatively charged. You just need to write those only examples. Structures are not necessary in classification part. Next, amino acids with positively charged group. Positively charged in the sense because amine is present and amine will accept a proton. Because nitrogen has lone pair of electron, it will become NS3 plus. So NS3 plus is present here, is present here. That's why the entire side chain is positively charged. The entire side chain is positively charged because nitrogen lone pair of electron, it accepts a proton, becomes NS3 plus. Clear? That's it. This is all about the classification part. In the next video we will discuss its chemical nature part. Till then, thank you.